Welcome to everybody. My name is Danielle Kashubi, and I'm the president of the Western Bird Banding Association. I'm going to share my screen so you got to see my face for a few moments there. But these webinars are part of the Western Bird Banding Association's webinar series. We try and do this once a year because we don't all get together and hear about what everybody's doing. We can't go to all the meetings. So this is our way of reaching out and hearing what everybody is doing. I'm looking forward to tonight's webinars, but we will be having webinars for a few more months. We're just doing them once a month. Next month, I am going to do a WRP on plumage workshop, and that'll be on February 11th. If you attend this webinar, I will send out the registration information for that, so you'll get to join that if you'd like to. We will have in March presentations by our Western Bird Banding Grant Award recipients, Lauren Hill and Alex Belladron, on their research. And then in April, we're going to have a presentation about barred owls and spotted owls by VTech. And I'm not going to try and pronounce his last name till I talk to him in person, but he's going to do that and in April and give us that presentation. So look in your email and social media for dates and Zoom links as we confirm those. These webinar series, the idea is that these support WBBA's grant program. We're proud of how these webinars build our community and share information. So if you want to consider donating to WBBA's grant program and support banders that are doing research and monitoring, and if you know somebody who needs a grant, tell them to apply. The information is on our website. This just takes you to the donation page, these QR code and that link. I'll bring them up again at the end so people can reach out if they want to. But if someone does need a grant for the research program, please tell them to apply. We do try and support research. We give out up to two $1,000 grants per year. And if we get enough support, we can do two $1,000 grants for people who are doing this research, especially people in early careers. This is really helpful. So Victoria is a native Texan who has migrated around the country in her pursuit of her passion, studying and conserving birds. We all understand that and love that. She's a proud alumnus of the Klamath Bird Observatory Banding Program. She not only earned the NABC Bander certification, but she earned a hallowed KBO Bander nickname, which she may or may not indulge us by telling us about later. Victoria's interests in molt gained in Victoria's interests in molt gained from many long hours of banding sparked her master's thesis question, what is the evolutionary driver of delayed plumage maturation in painted buntings? After her time at the University of North Texas Grass Lab, she intends to first pursue the NABC trainer certification, then plans to further her conservation mission in the service of a nonprofit or enrich her knowledge of avian life history strategies in a PhD program. So I'd like everybody to welcome Victoria tonight. And Victoria, you have the floor.
Okay, hi everybody. Let me get my screen set up here. And in the meantime, I guess I'll entertain you with my nickname. I earned the nickname Black Cap Chickadee because I am small and relentless, according to my boss, which is an honor.
I
Um,
think that's perfect. Good name.
Thank you. Okay, so let me start officially. Hi, everybody. I'm Victoria Langham. I'm from the University of North Texas in Denton. So that's all my fault. That's where everybody's coming from. I'm a master's student presently, and I'm here to talk to you about delayed plumage maturation in painted buntings. So here's my introduction. This is the painted bunting. They're a beautiful rainbow-colored songbird, or at least the males are. They breed in the American South each summer. They have a habitat of prairies and grasslands. They eat mostly seeds. They nest in open cup grass nests. They forage in low shrubbery or on the ground, and they are relatively stable. However, keep in mind they're a grassland bird, so they're still at risk. And then next to our beautiful adult rainbow male, I have a picture of a female or suspected female. We'll get to that in a minute. Before we learn about delayed plumage maturation, we'll need to learn a little bit about the painted bunting life cycle. I just want to check real quick. Are y'all seeing the Zoom sidebar or not?
The Zoom side. No, we don't see that. We just see your screen and
Okay.
So you can move that zoom sidebar though if you wish to get it out of your way.
I think I will. Okay. All right. Back to the presentation, everyone. Thank you. I didn't want my pictures to be blocked. Okay. So painted buntings hatch from these beautiful eggs in these grass cup nests. And when they grow in their juvenile feathers, they look like this. Very fluffy with a yellow gape, and they have little lemon wing bars on their wings. Then, in about August or late July, they'll molt into their formative plumage or their teenage plumage. That's the next plumage in their cycle. And you'll notice something that the boys and the girls look identical in this stage of molt. About next summer, after they have finished their first adult breeding season, they'll molt into their definitive adult plumage. Now, this is definitive, meaning that they will be in this same plumage for the rest of their life. So they'll just get to molt in a fresh coat of feathers every year. Males will be this gorgeous rainbow color, whereas females will get a fresh coat of green with one added tract of green feathers instead of brown feathers. Okay, so let's define delayed plumage maturation. This is the absence of adult characteristics until the definitive adult plumage. Most birds, as soon as they are sexually able to produce, they will advertise that physically. Think of a cardinal and other famous species of birds. On the left, we have a delayed plumage male. He does not have any blue on him whatsoever and no hint of rainbow plumage. In fact, if I were to look at him with binoculars, I would suspect he's a female. On the right, we have a male not showing delayed plumage maturation. He's molted in a little bit of blue on his head and around his cheeks, and if I were to see him while birding, I would in fact be able to tell that he is a second-year male bunting. Okay, so we don't really know why DPM evolved. There are some hypotheses as to the benefits of DPM. I'm going to go over these now. Our first one is status signaling. This says that the male is conveying his naivety or inexperience to an adult rainbow male. So if he accidentally oversteps territorial bounds or he thinks he can get a free lunch, this conveys to the adult male, hi, I don't know what I'm doing. Please go easy on me. Maybe just scold me instead of chasing me out of your territory. Our second hypothesis has to do with female mimicry. This says the green plumage that resembles a female may actually allow the young male to be mistaken for a female by rainbow males. This would mean that the male could benefit by not acting overtly male. So he may forego singing and he may creep through the territories and sit in the trees silently, much like a female. Then our third hypothesis is crypsis. This one's really easy. Being green makes you the same color as the leaves on a tree. So this both hides you from territorial males, but also makes you a less likely target to get eaten by a hawk. All three of these hypotheses do have one thing in common, even though their mechanisms are slightly different, and that is all three of these hypotheses could help these males avoid aggression from older territorial males in some way, shape, or form. So I am conducting field experiments that allow me to test some delayed plumage maturation hypotheses. However, I'm not going to test the crypsis hypothesis in my thesis study. That's because you can never completely rule out that being the color of a leaf may have some side benefits. So before we talk about my predictions, I want to go through my experimental methods. That way my predictions hopefully make more sense. So first, I capture the birds. I conduct passive and baited mist netting every summer. I conduct this in the morning between sunrise and about 9:30 a.m. when it's relatively cool here in Texas. When I do passive netting, I open about 10 nets and I check them every 15 to 20 minutes because though it's cooler in the morning, it's still hot. And baited netting is the same concept, except with 3 triangular nets set up around a bird feeder, and I am hidden off camera in the bushes watching the net constantly. So I'm able to extract the bird once it gets caught. After I capture the birds, I put a silver USGS band on them, and I give them 3 color bands that make a 4-color unique band combination. That way I can tell Bob from Fred apart in the field. And I take some standard banding data you may be familiar with. I age and sex them, I take some measurements, and then I weigh them. I also take some catalog photos just in case. They have a lot of cool plumage variation. Okay, so my experimental methods. Now remember, delayed plumage maturation and the hypotheses deal with how young males and females look alike. So my test subjects are only going to be young males or females. So first I'm going to take a green bird and I'm going to put that green bird temporarily in a specially designed open air cage that has a GoPro attached to it. Then, now our bird is in the cage. I'm going to take this cage and elevate it on a stool so that it's not obscured by tall grass and it's safe and it is easily spotted. And I'm going to take that to a rainbow male's territory. Shortly before I hide in the bushes, I'm going to put out a speaker that is broadcasting Painted Buntings song. I hide in the bushes and I wait for the after second year male to show up. Here he is. I couldn't make him rainbow. He just had to be blue. Um, and as soon as he shows up, I stop the playback, but this is a free sticker, so I couldn't remove the music notes. Once I stop the playback, I trigger a button on my phone that makes the GoPro record the behavior of the green bird in the cage to use for later, and I start a 10-minute behavioral observation focusing with my eyes only on the after second year male. And I do this using a really cool app on my phone called Quick Capture by ArcGIS. This takes a really fast little survey point. It will record whatever I set up the button to record in addition to a GPS point and the timestamp down to the second, which is pretty cool. And I'll make this funny joke. My data collection is just this, which is pretty cool and helps me to pay attention closely in the field. Okay, so I'm considering 9 behaviors in my experiment, and these are all behaviors that are performed by the rainbow male. Keep that in mind. So I have the arrive button just to signal when he shows up, and I have some behaviors such as approach, which is coming within 3 meters of the cage, attacking, which is not attacking. The bird in the cage is safe. It's simply swooping within half a meter of or perching on the cage. I have call and song buttons for when the ASY male is chipping or singing. I have out of view when I can still tell he's there, but he's hidden behind a thick branch. I have leave for when I can see him fly off into the distance. That ends the experiment early. And then I have a perch button for when he's sitting there looking. Okay, so now let's get into my predictions. If my data supports status signaling, I'm going to see a very certain pattern of data, but if it supports female mimicry, I'll see something else. So if my data supports status signaling, let's pay attention to this pink graph right here, which will be the girls. If a rainbow male is seeing a female in the cage and he can noticeably tell it is a female, I would expect him to perform singing or displays much more often than I would any other behavior. But I just put attack here for a placeholder. By the way, I forgot to explain my display button. That is a combination of singing and wing fluttering, which is how they do their courtship dance. However, if I'm an ASY male and I see a boy and I can tell that it's a boy in the cage, I might be doing something called a warding behavior, which would be chipping or approaching the cage to get a better look, maybe swooping over the cage. All of these would be suspicious active behaviors. And I wouldn't be singing as much because I don't want to impress this male. I want to get him out of my lawn. However, if my data supports female mimicry, I would expect that the rainbow male would treat green males and females the same way, either singing equally towards everybody or performing warding behaviors towards everybody, just because he can't tell who's in the cage. So let's go over some analysis. I did actually collect data. I have had 1 field season out of 2 so far, and we can go over the data I collected now. So how am I analyzing this? I'm analyzing this data using something called a discrete choice model. It's a model used in economics, but I'm taking it and I'm able to modify it slightly for use in biology. So a discrete choice model analyzes and predicts how an individual makes choices from an infinite, replaceable, and mutually exclusive set of behavioral choices. Now, why did I say infinite and replaceable? Well, because this is an economic model, oftentimes it models things like resources, like food, which are finite and unreplaceable. If I eat an apple, it's gone and I can't eat that same apple again. But since I'm modeling emotions, I can always choose to be happy, or I can always choose to be to sing with no limit on how often I can make this choice. I will be doing this model in RStudio, using a package called mlogit. This is just a special package for making discrete choice models. And my model looks like this. Now don't worry, we only need to pay attention to two tiny parts here. This orange part is my response variable. This is the behavior chosen by a rainbow male in that moment. So in a 10-minute behavioral observation, each time I see a behavior, that is a miniature discrete choice model happening in the rainbow male's mind. So each time he chooses to sing, he's making a choice in this model, which I think is pretty cool. And then this blue part right here is my individual level predictor variable. This is the sex of the bird in the cage, so either a boy or a girl. And this predicts how my rainbow male is going to react in the moment. Okay, I do have some preliminary results. Let's go over them. So most commonly, rainbow males chose to sing, be out of view, call, or perch, and they rarely chose to attack or perform a display. As you can see, they chose these last two choices so infrequently that they don't make up a decent slice on the pie chart. My model did return back some coefficients for this year. I have some negative coefficients and positive coefficients. Negative coefficients for this model simply mean that a resonant ASY male or a rainbow male is less likely to choose a behavior when he sees a male in the cage. So what are they likely to do? Let's look back at my negative coefficients. So a rainbow male is less likely to call, perch, sing, or remain out of view when he sees a second-year male in the cage. What do all four of these behaviors have in common? They're all stationary behaviors, as opposed to this positive coefficient, the attack variable. Now, a positive coefficient would be the reverse, right? A male may be more likely to choose this behavior if he can tell a bird is in the cage. However, I want to point something out. All four of my negative coefficients have these three little asterisks on the top, but my positive coefficient, attack, does not. My negative coefficients in this model are actually all statistically significant, which means my model is predicting with a certain level of confidence that a rainbow male will choose these behaviors in the presence of a green male. However, attack is not statistically significant at the moment. This means my model is not confidently predicting that attack is a choice a male would actively choose when he sees a green male. Now, just because it's not significant now doesn't mean that it could or could not be in my second season. I do want to highlight that this is a preliminary result section. However, we have modeled what a rainbow male isn't doing. So let's go back to what he might be doing. Perching, singing, and being hidden behind a leaf are all stationary behaviors. So we could infer that territorial males are more likely to choose active behaviors when they see a green male in the cage. These could be things like approaching, chipping, flying over, displaying, or attacking. They're not yet specific enough to make the inference. I have seen anecdotally two instances of a attack, both cases where a male swooped over the cage just as a little pass by. And I saw only one instance of a display, which was too few observations to be modeled. So I'm looking forward to gathering some more data next summer, and I hope that the volume of my experiments will make my model super well fleshed out, and we'll be able to model what males are doing instead of just what they aren't doing. But models are funny like that sometimes, and so is science. That is my entire presentation. Thank you for listening to me. I'm really excited to be here. I included my business card just in case anybody thinks of a question after this webinar. Please reach out to me. I'm happy to answer. And then I have my favorite picture from this past field season. And I'm happy to take any questions.
Thanks, Victoria. That was great. When we're done, can you put your email address in the chat just so if someone doesn't get it down right away, they can grab it from the chat.
Yes, I will.
In the meantime, does anyone have any questions? Feel free to unmute or write it in the chat, whichever you prefer. Let's see if anyone has written a question. No, not yet. So if anyone has a question, unmute or write it in the chat.
I had a question. super interesting analysis so far, but I was wondering what is like the mating strategy for the painted buntings, like monogamy, polyandry, polygyny, and is there a chance that there's a um, they have the delayed maturation for increased breeding opportunities as like a sneaker male?
That's a super awesome question. So, socially, as far as we can tell, they are monogamous, but that doesn't mean they're genetically monogamous. It is widely hypothesized that the green males could be sneaking in extra pair copulations behind the rainbow male's back, so to speak. That is something that is mentioned normally in the conversation of female mimicry. It would be super cool to do a genetic project on this data, but I did not get to have the funding for it, so I just did a field trial. I'm hoping that in the future, someone could take detailed blood samples of both parents and then nestlings, and then maybe we could get some insights on that.
Awesome, thank you.
Awesome. There's a question from the chat as well, and that is any thoughts on whether the cage itself might affect how the after second year male responds?
Ah, yes, and don't worry, I planned for this. I figured it would be boring in the presentation, but in my research proposal, I had outlined a certain number of empty cage trials. I also use an audio lure, as you saw in my methods. So I also have empty cage trials with no audio lure. You can imagine how that went. I waited for an hour and a half, and the rainbow male didn't show up. But when I did the exact same experimental protocols with an empty cage, in each of my trials, the rainbow male would show up pretty quickly, and the longest he stayed was about 2.5 minutes. And there wasn't any sort of territorial reaction upon seeing the cage. There wasn't song. It would often just be show up, a couple of chip notes, look around, and then leave, which I found pretty interesting. So I hope that means and my committee thinks that means the cage does not have an effect on how the territorial male responds. It's simply the bird in the cage that is eliciting a reaction from the adult male.
Another question from the chat. How many instances of a single behavior do you need to run the model confidently?
Hmm. I don't know the exact number for this. I know that I am treating my model, each instance of a behavior in a 10-minute experimental observation is actually its own mini experiment. So, according to the model, I need about a sample size of 30 for it to be flexible and confident. Normally, people would think that means 30 experimental trials, though. So I don't know whether to answer if I need 30 instances of a behavior or if I need 30 minutes of a trial. But I will tell you that my data set before being cleaned had about 4,000 behavioral observations to be modeled. So that's exciting. I could have almost a 10,000 choice raw data set, which would be pivoted into a nearly 50,000 row data set after the 2 years is over.
Awesome. I have some questions, just basic biology of this. First of all, how many, like percentage wise, of the population do the males do this delayed plumage maturation?
It's pretty common. Most of them are a majority green. I would say that a majority of the males are all green. However, it is decently common to see a male with a little bit of blue on his body, such that I don't really have a elevated reaction when I see that. I do think it's cool. But I have never seen a completely blue-green male. It has normally just been about half of the head is blue, and that's the most that I've seen thus far.
Okay, following on that, do the delayed plumage maturation birds sing at a different rate than the ones that have more blue on them, or do you know that answer?
That has not been specifically tested, but it is pretty uncommon to hear second year males in general singing. We
Okay.
do stop and ID any bird that we can hear if we can find it, and almost always it is a rainbow male singing. Occasionally, a few birds in my history on the project, I was also a technician before I was a student, so I was here for a little while. In about 2020, I think we had one green male hold a territory. It was pretty cool. And then since then and this summer, I have not seen in the records of another green male successfully holding a territory and reliably singing.
Cool. And do you do your testing in the cage with birds that do not have that do have blue on them, or just straight green birds?
Straight green birds. I am
Okay.
limiting, so that male with the blue all over his head, that is actually in my proposal as a bird that I would not use because I think it would confound the experiment.
Okay. Cool. Now I've asked all my questions. Does anyone else have any other questions? Okay. Thank you very much, Victoria. I appreciate your time.
Thank you.
Um, if someone else has a question they think of as they go along, feel free to write it in the chat. And Victoria, if you see them, you're welcome to answer, or we can ask them at the end. But thank you very much for your talk. It was great.
Thank you.